So I'm here representing our national service, which is the Genetic Health Service of New Zealand. I'm based here in Auckland, but we have a hub in Wellington and a hub in Christchurch, so we cover the whole of the country. We're a national service, so although we don't always achieve it, our goal is that we do talk to each other and we try to do things the same throughout the country. It's a goal that we always try to achieve. We don't always get there, but we hope that our service is sort of a model that you can, do the same thing for the same people irrespective of where they live in New Zealand. Yeah. So I did this talk, I think, almost 10 years ago, and I thought, right, what's changed? Well, not too much has changed. A little bit has changed, and we do a lot more testing, and we'll talk about the reasons for that now. Claire has already alluded to it. The other thing that's changed is that all the other speakers who have already spoken today have stolen, not my slides, but essentially presenting information that previously maybe a geneticist would only really talk about. But nowadays what we're seeing is that because genetic knowledge is expanding and it needs to get into all the different specialties, that a lot of what we would primarily do is now being moved out into the specialties, which means that more people are kind of getting the stuff that they need in terms of genetic testing and genetic counselling and stuff, but we still are important and we do have a role, and I'll talk about that specifically today. So this is the outline of my talk, and hopefully by the end you guys will know kind of the answers to all of these questions. I'm going to talk about the diagnosis of tuberous sclerosis, which has already been talked about a lot. Why do genetic testing? What can genetic testing tell us and what won't it tell us? Big questions that families will have if they have a child with TSC. They might say, well, can I have another child with TSC? What about my child when they grow up? Will they have a child with TSC? What about my sister, brother, uncle, aunt? They all want to know. Everyone is coming to us and asking us. So hopefully you'll know the answer to that by the time we finish. And the big thing that I will focus on, I guess, today is about reproductive options. Always we just present the information. It's important that people know all of this. It's not about telling people, directing people as to what they should choose. It's just about arming you with the knowledge so that you make the right decision for you, your family. And then maybe touch on the screening recommendations, but I know that's going to be talked about later. So very briefly, we'll do a little bit of basic biology science. Oh, that's not supposed to happen. Oh, that's a weird pointer, so I'm not going to use it. Okay, essentially, this is a stolen slide, obviously, but just talking about DNA, the molecule of life. So our body is made up of cells, and within almost every cell of our body, we have a nucleus, and inside the nucleus we'll have a copy of our nuclear DNA, so it's sort of like our instruction manual for how the human body is developed and how it works. So the DNA is packed very tightly into these structures called the chromosomes, and if you were to unravel them like this picture is doing, you'd see these, everyone's seen pictures of DNA before, this characteristic appearance of the double strand. DNA is essentially just a long billions of pieces of code. We only have 4 letters in the DNA alphabet: C's, A's, T's and G's. And a gene is, what we're going to be talking about, the basis of what we've been talking about today, the TSC1 and the TSC2 gene, is a defined piece of DNA which codes for a particular protein. So I sort of liken, when I explain this in my appointments, is that you can think of the human genome being like a really complicated book, and the chromosomes are like the chapters in the book, and the genes are the words in each chapter, so you sort of get down at the level, and then the DNA nucleotides are the letters that make up the words. So that's sort of a conceptual way of thinking about what we're actually doing when we're looking at genetic testing. So again, if you zoom in even more closely and you think about the gene itself, so here on the side closest to me we have the gene. What does a gene do? Well, basically a gene is like a recipe for a particular protein. That's the end product at the end. And we know lots about messenger RNA after the COVID vaccination education programs more recently. So essentially the messenger RNA will read the gene and then goes out of the nucleus, goes to a structure called the ribosomes, and then the ribosomes produce the code, the protein based on the code of the gene, and you end up with your protein. Proteins are very important for all of those pathways. That mTOR pathway is just one of them, but there are billions of different pathways in the body, and essentially it's all made up of proteins. So I think it's important to just sort of dwell a little bit on the terminology that we use now when we're talking about the things that we find on genetic tests. So we've sort of moved away from using the word mutation. There's sort of very negative connotation with mutation, and the big sort of development in genetic testing and genetic test results and things is that we now doing so much more testing compared to 10 years ago in all genetic disorders, and obviously the Human Genome Project has been mapped, and people can do all sorts of different tests, and you can look at your whole of your genome or your exome through a test, and we now know so much more about variation in the genome. And so we've developed sort of some terminology which helps us to understand when variation or variants are disease-causing or pathogenic versus when they don't cause any problems and they're benign, and benign in the true sense is that they don't actually cause any problems, and in this middle group where they're uncertain. And we have, we spend a lot of time now discussing results of uncertain significance to families, and just the importance of clarifying that and being careful about what your genetic test result is actually telling you. So a genetic variant is any change or variation in a gene. So a genetic variant may lead to a change in the way that gene functions, and therefore it causes a problem with that end protein. It might mean that protein is formed differently and it doesn't kind of fit together in the pathway like it should, or it might mean that there's none of that protein formed at all, so it's sort of a deficiency of that protein. So instead of the word mutation now we tend to say pathogenic variant. I'm not sure if that's any better, but that's what people have decided. But instead of saying we have a mutation in the gene, the technical term we would say is a pathogenic variant. So a pathogenic variant in a gene we would say is causing a genetic disorder. So some alterations in genes will have no effect on the gene function. And now that, thousands, probably millions of people have had their genomes sequenced, we know that there are so many variations in genes. We're all very different. Obviously, we're all very common in our DNA, but we have lots of differences as well, and most of those differences don't cause genetic disorders. So it's very easy. It's very common to do a genetic test to find a difference, which is probably not causing any problem at all. Then we have a difficult group in the middle where we find a difference or a variant in someone's gene, whatever gene we're looking at, but we don't know what it means, so we call that of uncertain significance. That might be pathogenic or it might be benign, and when you're thinking about whether your genetic test result is explaining your genetic condition, you need to be very sure that you know which one you're talking about, because you don't want to get it wrong. If you've got a genetic test result that's actually not the cause of the genetic disorder, and then you go and use that genetic test result for some of the things that we talk about later, you get yourself in a very deep, dark hole. Genetic variants can be present in our germline, so that means like after sperm and egg meet, there'll be a variant there from the start, so it's in almost every cell from then onwards, or you can acquire genetic variants. And so if you think about cancer as a bigger disorder, That's because as our cells divide throughout our lifetime, they may sort of collect these variants, which can lead to abnormalities just in a particular organ. So how do we diagnose TSC? Well, you've seen different variations of this diagram already. It's obviously a multi-system disorder. Sometimes it's very straightforward to make a diagnosis. So if you have, like we heard before with Helen, a child presenting with seizures and then someone noticed the skin marks, or they go and get the MRI, and then you go, okay, well, there we have the diagnosis, we made it in a day type thing. But it's not always that straightforward. As we've already heard, some of these features are age dependent, so you're not going to see another feature like in the kidneys and that kind of thing until that child is older. Some children won't present with seizures early on. They might present later on with developmental delay or some of the neuropsychiatric disorders, and then maybe someone will then notice the skin findings. So a diagnosis might come at different times in a person's lifespan. More and more we are seeing people who are adults who might find that they've got AMLs in their kidneys, and the question we get from the renal physicians or the urologist, does this person have tuberous sclerosis? So they then sort of have to go through a process of looking through all of these different body systems to make a diagnosis or not. So these are the clinical features, and you can make a clinical diagnosis based on clinical features, either by physical exam or special imaging or special assessments like seeing an ophthalmologist and that kind of thing. So this is a picture of the chromosomes. It's also important to think about how you can diagnose tuberous sclerosis based on a genetic test. So we have 2 genes involved in tuberous sclerosis, TSC1, which is on chromosome 9, and TSC2, which is on chromosome 16. So this is a different version of the pathway that you've already seen. So obviously we're interested here in the TSC complex, the 2 proteins that the 2 different genes code for. And we know that, here's the mTOR pathway here. It sort of regulates that pathway. It keeps kind of the brakes on it. So the way that I think about it is, if you live in cities, or you'll be very familiar with our on-ramp motorway lights that, let people get onto the motorway in an orderly fashion. So you can kind of think about the regulation of the mTOR pathway being a bit like that, and the lights that let the cars onto the motorway are kind of like the TSC complex. So when it's working, it says, okay, I'm just going to let you go on slowly, 1 car at a time, and everything flows along nicely. So it's like a negative regulation. If you knock out those lights, turn them off, then suddenly all of the cars are going onto the motorway, you're getting traffic jams, you're getting chaos, you're getting car crashes, that kind of thing. This is a very simple way of sort of thinking of the down regulation of a pathway, what happens if you don't have that. So as I said, you can diagnose tuberous sclerosis with genetic testing alone. Most people don't just go to their doctor and say, can I have a test for tuberous sclerosis? So there's usually a reason why you would have that test. And you might already have a diagnosis, and then you have a genetic test to say confirm it. We'll talk about that a bit later. So clearly, if you do a genetic test and the test comes back and says, yes, we found a pathogenic variant in TSC1 or TSC2, then you can be pretty confident that person has the diagnosis. There are very complex systems that I understand mostly, and scientists understand very well, about how we classify genetic variants. So if you're using a lab that's got a good reputation, you know how the variants are classified, you can be pretty confident that if they say it's pathogenic, it probably is. If you get a variant of uncertain significance, which means that yes, we've found a difference in the gene, but we're not entirely sure how that's impacting the function of the gene, then it's less certain. You can't say, well, this person's got features of TS and the genetic test has got a variant of uncertain significance. You can't automatically say that that genetic test result is causing what we're seeing in the person. And that's where often we get more involved in thinking about how can we clarify that. important to understand that even with genetic testing, this is changing all the time with our ability to do more deeper sequencing of genes and understand genetic variants better. But not often, but in some cases, you will find that you do a genetic test, even when someone's got a clear-cut diagnosis of TSC and they get a negative or normal genetic test. So you can't, in that scenario, if you've got a child with a clinical diagnosis and we do a genetic test and you get a normal negative result, doesn't mean they don't have TSC. It just means that you haven't found the cause. Yeah. So why do genetic testing? Well, there's different reasons why we might do it. The big change perhaps from 10 years ago to now is that in many cases genetic testing wasn't done because there wasn't really it wasn't going to lead to a change in management, or it wasn't going to make any difference. And it used to be quite an undertaking because it was done before sort of a new technology of testing was available. The genes had to be looked at one by one. It cost a lot of money, and it was sort of like, why would the public health system pay for this test if it actually is just going to tell you something we already know? So nowadays we would usually do it to confirm a clinical diagnosis, but it is a bit academic in some cases because you don't really need it if you've made a secure clinical diagnosis. If you have, tumours in the brain and AMLs in the kidney and the skin findings, you've got TSC. There's nothing else that can cause it. Yeah. Probably one of the biggest reasons to do genetic testing is to enable testing of other members of the family to see if they are at risk of the complications. And the kind of term that we use for that in genetics is predictive testing. So that's perhaps someone who maybe doesn't have features of TSC but wants a test, maybe a relative, or if you're talking about testing pregnancy where you don't know whether the fetus has it or not, that's sort of predicting whether or not they might have it, which is different to diagnostic testing where someone has the features, you're pretty sure you're not going to get a surprise. It's like, yep, you've got it, we're probably going to find it when we do the test. And so predictive genetic testing is done for different reasons. If you have family members, once one person's diagnosed in a family, other family members want to know for their own health, do I need to be screened for all of the complications? For parents, it's often because they want to understand their chance of having another child with the same condition and to allow the different reproductive options that I'm going to talk about to be available. So we still do most of the genetic testing overseas, but now genetic testing is done by a technique called next generation sequencing. In the old days, as I said, you had to laboriously go through the gene, sort of one gene at a time. That changed, about 10, 15 years ago, and now actually the machines look at as many genes as you want. It spits out the data, and it's still very complicated, but you have the data there to be analysed much quicker. There are lots of commercial labs that do this, and we tend to choose a lab in Finland, of all places. But the difference now is that once we've perhaps identified it in one family member, if we know that we're going to be offering testing perhaps for prenatal testing or pre-implantation genetic testing, or maybe you have 10 siblings and so there's going to be a lot of testing done in the family in New Zealand, we can set the test up here. We usually use the lab in Christchurch, so they'll have a test for your family's variant, and then it's much more straightforward. Blood goes to Christchurch, they're very efficient, give us our result in a few weeks type thing, so it's great. Most genetic testing is done on the DNA that you can extract from your blood, so it's just a normal blood test. You just go wherever you would normally go. There's no special instructions. People always get a bit like, genetic testing, and people are ringing us up from the phlebotomy centre saying, we don't know how to do this, and just take the blood, put it in the tube, send it. There's nothing special about it. Just make sure it's from the person that you... Sometimes we might like to test tissue. If Claire didn't talk about any epilepsy surgery, but if there was a piece of brain or a piece of the AMLs from the kidneys, might be useful to test that if we can't find it in the DNA of the blood, which I'm going to talk a little bit about as well. Nowadays, a lot of testing for TSC is actually not done by our service. It's done by a neurologist, maybe nephrologist, but in the paediatric setting, most often Claire and her team would just request the test. That's a bit different in the days gone by. They have to come and see someone like me, and then we would order the test. As I already mentioned, genetic testing doesn't identify a variant in all individuals, and then the diagnosis falls back to clinical criteria, and that is the more old-fashioned way that we used to do it. So if we had parents in front of us, we'd say, right, you need to have a renal ultrasound. If we could, we'd get you a brain MRI. Probably never get a brain MRI anymore for healthy parents. Look at their skin. Some of you might remember being in the dark room with the wood lamp. Yeah. So you can see, like, if we have a genetic test, getting a blood test is a lot more straightforward than having to find a dark room in the hospital and the wood lamp that always goes missing. So what can genetic testing tell us? Again, I think we've already talked about how people with TSC are more common to have a variant in TSC2 than TSC1. Familial, so what I mean by that is if you have a family history of TSC, then you're more likely to have a variant in TSC2. We think that overall some of the variants in TSC2 are thought to be milder. Sometimes, as I said, you might not identify a pathogenic variant on genetic testing, and overall the clinical features, or the phenotype is the word that we use, for those people tends to be less severe, and that's because we think they're probably mosaic, and I'll talk a little bit about mosaicism. And then, as we've already heard, that in rare cases you can actually have a chromosome deletion which removes both TSC2 and a gene that causes autosomal dominant polycystic kidney disease. So for those people, they actually have two genetic conditions, completely sort of separate. And that's unfortunate for them. So mosaicism is something that as geneticists we like and get interested in, get excited about. So mosaicism, you guys have probably heard of mosaic, which is where this word comes from. There's a nice mosaic picture there of a cat that has different colours in their coat, and the different colours in, the coat is explained by, different genetic changes in the genes that control coat colour. So mosaicism is the term that we use to describe when there is two or even maybe more genetic changes within a tissue. So, you might have an abnormality in a gene, and only some of the cells of whatever tissue you're interested in, and we're talking about testing blood, would have that abnormality in it. So how that relates to TSC is that some people will actually be mosaic for their gene variant. So some, but not all, cells of their body will have the genetic variant causing TSC. And you can sort of, you can understand why then, if only some of the genetic abnormalities in fewer of their cells, why they may then present with lesser features. It's not always the case, but in general that's how it works. So now moving on to sort of inheritance of TSC. So you guys will all, I'm sure, know that TSC is an autosomal dominant disorder. So there are two different genes, as we've talked about lots, and you either have an abnormality in TSC1 or TSC2. It's not both of them. Some people get confused and think you have to have abnormalities in both. Autosomal dominant disorders occur when you have an abnormality in one copy of the gene pair. So all of our genes come in pairs, so we all have two copies of both of these genes. And for individuals with TSC, you would expect to find an abnormality in one copy of either TSC1 or TSC2. The other copy is still working fine, but for this you need both copies working normally. So a pathogenic variant can be inherited from a parent, or it might be de novo, which is another term that we use, which essentially just means it's a new, spontaneous, sporadic, random, whatever you want to use for that word, but it's a new onset. It's only present in the individual with TSC. It's the first time in the family it's popped up, that kind of thing. So that is the more common scenario that we see. So a child or an older individual with TSC due to a de novo pathogenic variant. So in about 2/3 of cases, when you're meeting that child, you ask their parents, you look at them, you look at their skin, and you go, okay, it doesn't, and you ask them about their family history, and it appears that neither parent is affected. So that's the more common scenario. So why does that happen? Well, when DNA is copied and put into the egg or the sperm, it's like copying, billions of pieces of information, and every now and then there will be a variation or a variant that, an error in that copying process. That's kind of how, as a species, we evolve, that our DNA varies as it gets passed on, and most of the time those new changes don't cause any problems. But if it occurs in an important part of a gene and it stops that gene from functioning, then that child will have a genetic disorder for the first time in a family. So even when we think that it's de novo, we've been called out more times than we would like to admit. We would examine parents. Maybe we need to image them, as I talked about before. We used to do that a lot, now not so much. But we would usually always offer genetic testing. If we do genetic testing and both parents test negative for the variant that's been identified in the child, so genetic testing would always start with the affected individual, and that's typically in this scenario we're talking about the child. We don't just test the parents, we want to know in the child first what it is, and then we can look in the parents for that thing. So if we do those tests and the parents are negative, we could estimate that the risk of recurrence for the next child would be around 1 to 2%. Now that's higher than a family that hasn't had a child with TSC, and why is that? There is a theoretical possibility that one parent may actually have the TSC1 or the TSC2 variant in either a small number of eggs or a small number of sperm cells, or maybe even in tissue, in cells outside of the gonads. So the term we use for that is gonadal mosaicism. And we're not going to start testing eggs and sperm, no way. We just say that's a theoretical possibility. It's actually been reported multiple times in TSC, so it is a real thing. And so that's why the risk is an estimate, and it is higher to say that yes, you could have another child, but still not very high. If after that process of doing genetic testing or examining, or just because a parent says, well, hey, yes, we do know people in our family have TSC, we then establish that actually the TSC1 or 2 variant is inherited and the parent has it, there would be a 50% chance of passing it on next time. Really, really important to understand, and we've already mentioned it before, that the condition can be highly variable. So if you have a child who has severe features, the parent might only have mild features. There could be someone else in the family that's moderately affected. And that is kind of a similar thing that we see in a lot of dominant disorders. And the reason we explain that is, well, why we think that is the case is, as you saw before with the pathway, is that the mTOR pathway is just one thing. There's so many other things that feed into it. And it's likely that all of the outcomes of all of those other pathways are influencing an individual's phenotype. It's not just the one thing in the one pathway. But we would say that penetrance, so penetrance is the word that we use to say if you have a genetic pathogenic variant, how likely you are to display signs of the condition is 100%. So some genetic disorders you might actually have an abnormality but never have any features at all. people say, is that, can things skip generations, or can you be a silent carrier or something like that? So TSC is not like that. Although someone may be very mildly affected, maybe they just have a few hypopigmented vacuoles, maybe some facial angiofibromas, just a few, and have never had any sort of medical issues. They might have AMLs in their kidney that have never given them any problems. They still have signs of the condition, they just wouldn't consider themselves affected, I guess, in a way. So you've probably seen these diagrams before. This one is taken from the TSC, TSA Australian site. So essentially it's just talking about, say, in this example, the father has TSC, and we use these little the mixture to say that they have a little d and a big D. So the big D is the faulty genes, the way they talk about it there, and in their sperm, on average half of the sperm will have a normal copy of the gene, and half of the sperm will have the copy with the fault. So that's how you get the 50-50 outcome. And then you assume that the mother doesn't have TSC, but in some cases people with TSC might get together and have children, and that would make this diagram a bit more complicated. So autosomal dominant disorders affect males and females. It's passed on equally between men and women. TSC doesn't really affect fertility, so it's not like, if you're there's no difference between passing it on between men and women. And it's 50% each time you have a child, so if you've already had a child with TSC, it doesn't mean that next time you're not going to, if you have 2 children, that kind of thing. Chance has no memory, it's like flipping a coin each time. So I talked a little bit about mosaicism, and that's what we probably have learned a lot more about in the last 10 years, is that it is actually far more common than we understood before about for a parent to be mosaic. And the reason we know that now is that our testing is better at picking up lower levels of pathogenic variants in genes. So if we then said that a parent was mosaic, so maybe, I'm not just talking about being mosaic in your gonads, your eggs or your sperm, I'm saying maybe in the rest of your body you have a very low level of the TSC1 or 2 variant, it's really hard for us to estimate what that risk of passing it on might be. This diagram here is just sort of showing, this is taken from just a generalised epilepsy website, because we now know that for many genetic epilepsies, for some reason there seems to be a higher rate of mosaicism in parents. The key thing to remember is that if the parent is mosaic and probably very mildly affected, if they were to pass it on, it would be non-mosaic in their child, so you would potentially expect it to be more severe. Okay, so let's move on to the reproductive options. So I think it's really important, as I alluded to at the start, is that when I present this information, is that it is just about providing the information to everyone. And it's not up to any medical specialist to decide which might be the best thing to do. I think it's important for people to hear this information at a time that it is relevant for them. When a lot of the time they're sort of on the list when people are being transitioned, children to adolescents to adult services, is to get genetic counselling. And we do get quite a lot of referrals for teenagers, and often when we meet that family and we delve into it a bit more, it's actually not the teenager who's worried about their children in the future. It's actually the parents worried about their grandchildren, which is fair enough. As parents, we worry about our children and we worry about our future grandchildren, but we have to remember that actually talking to teenagers about reproductive options is often not the best time. It's not relevant for them at that time. We're very happy to have conversations with parents, and that can usually be done in a less formal way, just to let them know that yes, there are options and this is how you might access them, and even in 10 years' time there might be even better options. So please just come back and talk to us when it's more relevant. Yeah. So the options I will talk to you about today is starting with, I guess, saying the option of not doing anything different after having genetic counselling is obviously appropriate for some people. This might be if they know that the risk is estimated to be low, or even where the risk is estimated to be higher, so maybe up to the 50%, but that the reproductive options available might not suit them for personal or cultural or religious reasons. So some people decide we're not going to do anything different. Or maybe their lived experience of the condition is such that they wouldn't change, the course. It's important that people actually understand what the risk of recurrence is, or the chance of recurrence, whether it be lower, bit higher, and that it's the same chance each pregnancy. To sort of chime in on Claire's really good talk about early diagnosis. So we would say, look, if we know that your child is at higher risk of tuberous sclerosis, we want to make sure they get diagnosed as early as possible for all of the reasons that Claire talked about. We would offer more detailed scanning in pregnancy, and that would usually include a fetal echo. That's the most useful thing if you can find the cardiac rhabdomyomas. Potentially doing brain MRIs in pregnancy is also may or may not be helpful. That would all be accessed through maternal fetal medicine service, so it's important to know about pregnancies, and so the right referrals can be made at the right time. Taking a step back for all of these different options, if it's the mother who has TSC, it's important that they talk to their specialist before they have a pregnancy, ideally, about the best way that their care might be modified with pregnancy, with the different medications that they're on, if they're having seizures, are there potential impact on the fetus? What if we stop medications? Is that going to be worse for the mother? So, it's complicated. It's good to plan pregnancies. Not all pregnancies are planned, but I think it's important for people to ask these questions and to make sure they get good answers about them. Once the baby is born, then someone should be looking at that baby and seeing whether there are any obvious features such as hypopigmented macules. Nowadays, because we hopefully have done genetic testing, or if we haven't and we know about it soon enough, we could do testing in the parent who's affected and then have a genetic test ready. We would have it set up in our lab in Christchurch, so taking blood from baby after 1 or 2 days, same time they get their other blood tests, and getting a genetic test result back, within a week type thing. Yes or no, does this baby have TSC? And then if no, then obviously that's great, you don't need to continue the monitoring, but if it's yes, then doing all the sorts of things that Claire was talking about. For some people, they are aware of the risks and they don't want to have another child with TSC for different reasons, and so they might consider sort of non-biological or non-genetic parents, and so adoption, sperm or egg donors, obviously always a possibility, and something that would be for sperm or egg donors accessed through fertility service. So the next slide I'm going to talk about is the reproductive option that we would term prenatal testing. So prenatal testing is just a term to encompass any kind of genetic testing in pregnancy. So this is done using one or either of these procedures. We would term these procedures invasive procedures because there is a small risk of procedure-related miscarriage. So the first and the earliest test that could be offered would be something called a CVS, which is under ultrasound guidance, where a small amount of the placenta is biopsied, and then genetic testing is done. This is usually only done if the couple undertaking the testing would consider changing the management of the pregnancy. You don't really need the information that early, just so that you know whether the baby is going to be affected. There are other ways that we can do that. As I said before, we can do genetic testing very soon after birth. Amniocentesis is slightly later in pregnancy, from 15 weeks, similar in that the procedure for the woman is the same, but instead of taking placenta, it's taking a sample of amniotic fluid. To have these types of tests, you really need to know what you're looking for. If anyone's had genetic testing before, you'll probably remember that it can take weeks or sometimes months to get the results. You clearly don't want to be in that scenario if you're testing a pregnancy. So this kind of test is looking for a known variant in the TSC1 or TSC2 gene so that we can get results quickly back to the couple. Prenatal testing like this is usually done through a MFM centre, so Auckland, Wellington, Christchurch, some exceptions, but it needs to really be planned. Obviously you need to have an appointment at the right time in pregnancy, but for genetic testing for a specific condition like this, it's good for our service to be involved and to pre-organise all of these things so everything runs smoothly. The other option I would mention is something called pre-implantation genetic testing, or PGT. So instead of having a pregnancy naturally, this would involve undergoing IVF. So as I said before, it's not usually a fertility problem for adults with TSC. The whole purpose of this is creating embryos in the lab so that you can then do genetic testing. And very simply put, this means that you test your embryos after about 5 days. You can biopsy. Down here is showing a day 5, what we call blastocyst, where the embryo is made up of 6 to 8 cells. You very gently suck a few cells out, and the DNA from that is then amplified, and you can do genetic testing. Again, this is not just doing generalised genetic testing. This is doing a test that's specifically designed for that couple, and you need to know what you're looking for, and you also, the lab that does this would have set up a personalised test for that couple. So this is not something that you just go along to your IVF provider and say, hey, let's do this and we'll start tomorrow type thing. Genetic counselling is the essential first step. First of all, we want to check the results to make sure that this is actually the cause of the condition, because if you get it wrong, you might be testing your embryos for the wrong thing. In New Zealand, there is public funding. The good thing compared to 10 years ago is that the waiting lists are coming down. We've always had public funding for PGT in New Zealand, which is a difference to Australia, because they're only sort of just getting into that space, which is good for Australia. You can also privately fund it. So I've pretty much run out of time because I just wanted to leave a bit of space for questions, but I just did include this because obviously when we're doing predictive testing, we would also always talk to people about what screening they need to access once they've had their test, if they come back as positive. So the summary is essentially all that I've talked about. Just to mention our service, we are a very accessible service. Not everybody knows about us, but if you do know about us, actually, you will find that if you phone and speak to us, you actually, there's not many health services in our country where you would get put through to a genetic counsellor who's highly specialised, will know probably more about the genetic condition than maybe many of the other health professionals that you've talked to before, and will probably know either how to answer your question or to help sort you out. Our genetic counsellors are amazing professionals, and they all want to help, and they know how to navigate things for people, and they often will take it on. So please just think about us and contact our service, and we might not be able to solve your problem, but we will hopefully be able to, direct you in the right place if we can't fix it for you. So I think that is my talk, and I don't know if there's any questions. Obviously some questions that you might want to ask me might be not appropriate for a public forum. So I can't stay for long, but you can always, Helen has my contact details, so if there's any sort of more personal questions, you can always get in touch with us through Helen. Thank you.